Okay, hi guys. We're going to talk through protein synthesis today, and more specifically how organelles work together in order to make a protein. So prior to this video, you should have some knowledge of organelle function and all of those involved in eukaryotic cells. Today we're going to talk about how they link together in protein synthesis. So just a quick reminder, DNA is transcribed into messenger RNA, which is then translated into a protein. This happens in the ribosome. Transcription happens within the nucleus itself and making the RNA from the DNA. So we start off with the nucleus. We have the DNA, and the DNA is unzipped by an enzyme called DNA helicase, which then allows us to transcribe messenger RNA by adding RNA nucleotides together using RNA polymerase. And that RNA polymerase is then going to leave the nucleus out of pores, nuclear pores, and be on its way. It's then got two choices to make. So if this RNA is going to be something that's going to be used within the cell, it's going to head away from the nucleus and head towards three ribosomes that will be floating around in the cytoplasm. So when it reaches this ribosome, amino acids are going to link together, forming the primary sequence of the polypeptide, and that is then going to give us a new protein, and that can be used within the cell. However, what we're thinking about today, a little bit more complicated than that, and it's what happens when a protein that's being made is going to be expelled out of the cell, a process called exocytosis, and that requires a few more organelles linking together. So let's start again with the nucleus here. Here is the DNA that's been unzipped. We have the messenger RNA that's been formed that transcribes the DNA, and then it's going to leave out of these nuclear pores. Now, in continuation with the nucleus, we have a organelle called the RER. Very similar to the SER. The one identifiable difference, apart from that it's in continuation with the nucleus, is that it has these ribosomes attached to it. So that's where rough endoplasmic reticulum comes from. It has this rough surface as a result of the ribosomes. So the messenger RNA that leaves through the pores ends up inside these flattened tubes, reaches a ribosome, and then what comes out is a polypeptide. So that polypeptide sort of stays within the RER, ready to be transported. So where it's going to head to is an organelle called the Golgi apparatus. And we should have sort of read about this prior and understand that the Golgi is responsible for modifying and packaging the protein. What does that mean? Well, what that means is it's going to add signaling groups that are going to tell and direct the polypeptide where to go and what to do. So it gets those carbohydrate groups from an organelle called the smooth endoplasmic reticulum. So this is another set of tubes, just like the rough endoplasmic reticulum, except it doesn't have ribosomes. So this is responsible for the synthesis and storage of lipids, like cholesterol, and carbohydrates. So they form molecules that are going to be transported from the SER to the Golgi. Now, some proteins, aside from kind of being modified and packaged with these groups, also require these groups for their function. So signaling proteins, for example, are often glycoproteins, and these will require a carbohydrate group as part of their quaternary structure. It's part of their structure that involves that enables them to carry out their function. So they get those or require those from the SER. But how does the protein and the polypeptide in the rough endoplasmic reticulum, and how does the carbohydrate and lipid groups that we've got, how do they get from where they've been made into the Golgi? Well, that's through a transport network called the cytoskeleton. And these are basically kind of tiny microtubes that act as like a railway network that get the components of the protein to the Golgi, where they are going to be put together, packaged and modified, ready for expulsion out of the cell. And we've got the same network that's going to transport the finished article, finished protein, to the membrane ready for exocytosis. So what happens here is the products that are inside these vesicles here bud off, and they form sort of smaller vesicles that are transported on this cytoskeleton. They deposit their contents into the Golgi. The same thing happens here. We get a vesicle snapping off the end of the Golgi, heading towards the membrane, and then we get a process called exocytosis occurring, where the components, or the finished protein, are going to fuse with the membrane and deposit the protein out of the cell, where it's going to move on. And as a result of these signaling groups that it's acquired in the Golgi, it can be directed and it can move towards the site of action where it's going to be required. So that's how a protein is going to be made from start to finish, how it's going to be expelled, and how all of these organelles link together, 1, 2, 3, 4, out of the cell.